Welcome back. Here we go. We're going to put this all together. Have a question for you at the end of this one. There are 2 more videos in this lecture series. These are the most dense ones. Make sure you've watched the other ones so that you have all the parts together. Pull out your picture of a chloroplast. Pull out your notes from topics 7 and this topic, and let's get started. There we go. Holy moly, this is a thylakoid, and that's all the pieces we had in the last lecture, the last time I videoed. Remember, we're doing linear electron flow. Light's going to strike the thylakoid. It's going to strike the leaf, the chloroplast in the leaf, the thylakoid in the chloroplast. It's really going to strike both of these photosystems. In each photosystem, we're going to get some excited electrons. We're going to use the energy in that electron, but we also have to replace the electron because we want this to flow. If we got an electron excited and it left and nothing ever happened again, that would be the end of the process. So we always are going to have to replace the missing electron. If something gets oxidized, we're going to need to reduce it in the next step. We're going to have a lot of redox reactions. So let's put it all together. Photosystem 2, remember we start with photosystem 2. Light strikes photosystem 2. It's striking everywhere, but we're going to try to make this happen, in a story. So we're going to start with photosystem 2 getting struck by light. I made up names for these kind of as a note. I broke this down so it was easier for me to teach. This is not the boost photosystem 2 electron is not the official name of this step. There is no official name. I just tried to organize them a little bit for you. So we're going to start by boosting an electron, getting it excited. And we do that by the photon, the little particle of light, striking the chlorophyll molecule in photosystem 2, causes it to wiggle because it's so excited, and they bump into each other, and this is like an excitement party. And ultimately, it strikes, here, it gets so excited, these are so much energy, it gets passed to this pigment 680, this actual set of pigments that are really good at absorbing wavelength 680. This is a molecule that's really good at getting an electron excited in response to this red-orange light. And so when it gets excited, it has an electron, leaves it. That means P680 is oxidized to P680 plus. It cannot continue to give up electrons now. We're going to kind of skip ahead and then come back. This excited electron is passed to a reaction center. We're just going to call it the primary electron acceptor. This is a part of the molecule here that is waiting for an electron. It is waiting to be reduced. The primary electron acceptor is reduced by the electron that left P680, and we're going to pass it on to the electron transport chain. Every time we pass an electron, when it leaves one molecule, that molecule is oxidized. When a molecule gains an electron, it is reduced. So as we transfer our electron to the primary electron acceptor, the primary electron acceptor is reduced. P680 is oxidized. As this electron is passed from the primary electron acceptor, this is the same picture but with more now. This electron is passed to this protein. You don't need to know its name. You can just say it's a protein here. The primary acceptor is oxidized, and this protein is reduced. The reduction of this protein causes it to pump protons, H positives, into the thylakoid space, the lumen of the thylakoid. So this electron, excited, is passed to this first protein in the electron transport chain. It causes, as it keeps getting passed, every time it's passed through these proteins, as this one loses an electron, it becomes oxidized. The oxidation of PQ is the reduction of CYT, and the electron, as it's passed, causes this protein to pump a proton through into the lumen of the thylakoid. These are proton pumps. They pump based on their oxidation and reduction. Now you'll note here that these electrons in the picture, it was kind of low, and then it got super high up in the picture, and then it's pretty low, it's getting lower and lower. The picture is trying to show you that each time this electron moves, it loses a little bit of energy, a little bit of entropy, a little bit of heat. This is it can't be a perfect system. It's not perfectly efficient. So this electron gets less and less potential energy as it's passed from protein to protein. But what it's doing here is it's generating a hydrogen gradient, a proton gradient. This, remember, H plus is just a proton. It's just a positive subatomic particle. And it's causing there to be a lot more H pluses inside than outside. So kind of as a note, throwback to topic 3. Maybe even topic 2. What do I call a solution that has a lot of H positives in it? It's an acid. So what's happening is these proton pumps are pumping, and they're creating an acidic environment, a much more acidic environment inside the thylakoid membrane in the lumen than in the stroma. Those H positives are now going to diffuse through ATP synthase. They'll just flow down their chemical gradient here, and it's going to cause ATP synthase to spin, and it's going to make some ATP. Again, I don't care how much, just the fact that it is making ATP. There's a lot of light. There's a lot of proteins on the thylakoid doing this. There's not just one per thylakoid. Keeping count doesn't matter. Just know that it's a lot. Lots of hydrogen flowing. Lots of ATP is being made. Somebody always raises their hand at this point in class and asks me where the ADP comes from. This has been produced by the cell when it broke down previous ATP. You do build the molecules in a different system we're going to talk about later as well. So just assume, if I don't give you anything leading in, like where did this come from, just assume that it is abundant, and I'm probably not going to explain to you where all of it comes from. There's enough ADP and inorganic phosphate to build as much ATP as this cell needs. So here, this is still photosystem 2. This is the first electron transport chain. We had an electron that was excited by light. P680 was oxidized. It became P680 plus. That electron was passed to the electron transport chain that pumped protons into the inner membrane space and ultimately went through on the other side through ATP synthase and generated ATP. But remember, I want this to flow. I want it to continue to happen. And P680 has already been oxidized. I can't continue to oxidize it to nothingness. I need to reduce it before we can do it again. I reduce this with water and the electrons from water. P680 is extremely strong at oxidizing water. That means water will split as a result of this reaction. As light strikes the photosystem, water splits, and the electrons from water replace the electrons that were lost by P680. Water is oxidized. P680 plus is reduced back to P680. The electron from water is transferred to P680 positive. P680 positive is reduced back to P680 with no charge. And oxygen is released as a byproduct. So as this happens, the electrons end up in this protein chain. The oxygen is diffused out of the membrane. Oxygen is small, it's nonpolar, it can diffuse through the membrane. The hydrogen ions get stuck. These H pluses get stuck But that's awesome because we're trying to create this really acidic inside the thylakoid system because we're going to pass them through ATP synthase. That's just going to make more ATP for us. Perfection. All right. So far, we have excited an electron. That electron was passed through the electron transport chain. It pumped protons as it moved, and those protons filled the thylakoid lumen and then passed through ATP synthase to make ATP. Every time that happened, we needed to replace this electron that had been oxidized away. So water is split. Water essentially forms an inexhaustible supply of electrons. Oxygen's a byproduct. The hydrogens stay inside and the thylakoid until they can pass through ATP synthase. And it just keeps flowing. It's like a turning on the tap. But now the problem is here, look at this electron. It's as low in energy as it was at the beginning of this process. It's actually a little bit higher, so it's not completely back down to what it was in water. But it's pretty low energy. And I'm not done. I want to do some more stuff with it. So we're going to have to zip it with light again. I put both the systems here. This is photosystem 2 and its associated electron transport chain. And here's photosystem 1 and its associated electron transport chain. We're going to strike with light again. Light is also striking photosystem 1. And when it does, it gets all these molecules so excited and they're bouncing around. And it eventually strikes this nice pigment center, P700, and it oxidizes P700 into P700 positive. P700 positive's lost electron jumps up to an electron acceptor. P700 is now, it has been oxidized. It's going to wait for another electron to come. The energy in this excited electron that we oxidized from P700, we reduced the primary electron acceptor, and this one is also called primary electron acceptor. We reduce it, and then we oxidize the primary electron acceptor by passing that electron to this molecule called ferredoxin. We are going to use this excited electron to pass it ultimately to an enzyme called NADP positive reductase. It's an enzyme, ASE. It reduces things. It's a reductase. The thing that it reduces is NADP positive. NADP positive is abundant in the stroma. Hydrogen ions that have been pumped through ATP synthase are now floating around in the stroma. These are the ones we got from water in the first place. NADP positive reductase takes the excited electron, NADP positive, and a proton, and it puts them together to make NADPH. These electrons are ultimately flowing from water. These hydrogens are the ones that had been previously torn off of other waters. The electrons and the hydrogens in NADPH originally came from water at the beginning of this story. This ferredoxin here, I want to sidestep a little bit. You can call it just FD for now. This is one of the earliest evolved proteins in this entire story. Ferredoxin has iron in it. It has a cofactor of iron. Iron is really a good catalyst. The iron-sulfur hypothesis, that's why we think iron is involved, because iron is very good at giving up electrons. In fact, in this process, ferredoxin is absolutely brilliant at giving up electrons. It's very good at handing them off to NADP positive reductase. This ferredoxin is an amazing enzyme that allows these redox reactions to happen very quickly. So with the whole picture here, mostly, we can't, we can't continue unless we have a way to replace this electron. Remember, this light struck the P700 ultimately and oxidized it to P700 plus. We're going to replace P700 plus's electron with the electron that started out in photosystem 2. Water was split, and its electron went down the first electron transport chain, and then it replaces the missing electron in P700 plus, and then it gets excited again. Remember, we're flowing the electrons. If I tell you this story from starting with splitting water, water's split, its electron is excited. Its electron is passed through the first electron transport chain, and that excitement is used to generate the powering of the spinning of ATP synthase, which causes the production of ATP. This electron is excited again, and now this electron is built into NADPH. It helps to reduce NADP positive into NADPH. It's still pretty excited. I don't know how it is up in the picture. And this molecule is now going to be a source of excited electrons. We made energy and excited electrons in a carryable form. I put the whole picture here. I don't want you to copy this exactly. I'm not going to ask you the names of PQ, CYT, and PC here. Photosystem 2 has an electron transport chain that is a series of proton pumps. Those electrons end up in photosystem 1, where they get excited again and are passed to ferredoxin and finally NADP positive reductase, where they are built into NADPH. Meanwhile, we made an awful lot of ATP. With that, I'm going to leave you off. Make a big picture. We made ATP, NADPH, and oxygen. We don't care about the oxygen, it's a byproduct. Write down, before you start the next lecture, write down the formula from photosynthesis from the very beginning of this lecture series. We're missing something. We have a whole molecule in there that's kind of important that we haven't used in our process yet. Circle it, because that's what we're going to talk about next. And I'll see you in the next video. All right, bye-bye.